try to write it down, and if you can record, you record it, because the material that I prepared initially was like a guide so that students can go through, but it's a skeleton. So I am supposed to give you the flesh. It's just the basis. It's a summary. All the material I sent to you is a summary or some of the topics. Those are not the main notes. So usually I do not have a note as we come to class. So what I teach you now, if you ask me to teach you again in the next 1 hour, it's not be the same thing exactly, but I will still explain the body, because like one of my mentors in the university is retired now. That's how he teaches. He has the experience, everything. He just come to class with some things to serve as guide. So when he's talking, students will not say, please sir, I missed what you said. Can you take it again? He said, well, I didn't even know what I said. Ask your friends. And the reason is because he's not reading from a note, but he's just looking at the summary as guide and telling us what he is saying. So what we want to look at today is laboratory methods of assessing the microbiological status of different classes of food. Laboratory methods of assessing the microbiological status of different classes of food. There are different classes of food. If you remember from primary school, we have carbohydrates, proteins, vitamins, mineral salts, fat and oil, and water. So these are the 6 classes of food, and there are laboratory methods of assessing the microbiological status of each of them. The way you will examine water is different from the way you examine fruits, and it's different from the way you examine rice or beans in terms of the microbiology. The way you examine cooked rice is different from the way you examine uncooked rice. So these are some of the things we're going to look at today. But let me just say that I want to talk about laboratory methods. Thank God that towards the end, we now have microbiological status of different kinds of food. What we're talking about, the laboratory, which laboratory? There's no laboratory in engineering. So the laboratory we're talking about here is the microbiology laboratory. So this is just saying microbiological laboratory method of assessing the microbiological status of different classes of food. We say microbiological status because the contaminant or the spoilage organism in the food can be bacteria, it can be fungi, it can be algae, it can be a protozoan, and it can also be a virus, and it can also be a nematode. All these are microorganisms. So most times, if assuming it's only bacteria that is involved, we just say bacteriological status. If it's only fungi that is involved, we could just say fungal status. If it's only virus that is involved, we can just say viral status. But because it's more than one type of microorganism, so we have to cover it up by saying microbiological status. So that tells us exactly that it is not just one microorganism. So don't go ahead and be saying bacteria or mentioning a specific when you are trying to explain. So you should know when to differentiate and when to use collectively. So several laboratory methods are available to assess the microbiological status of different types of food. And the methods are many. Let me just take this before we go on, that in examining the microbiological status of a food, there are certain principles that you need to take note of. Now, let's look at this method. Let's look at this list of methods for enumeration methods. I want to list enumeration methods. We want to enumerate. If you check dictionary for enumerate, means you want to count. You want to list. So enumeration methods include the following. Number 1, plate count. You can even see from the word count, you count the plate. It's not the plate you count to, it's the microorganism on the plate that you count. It's a pity I don't have Petri dish with me here. But when you have a Petri dish, you can Google what Petri dish means. Petri dish, this is a bottle of a drink. But Petri dish, this is what I can find here now. I don't want to stand up and go and be bringing this thing from there. Petri dish basically means Petri dish. When we say dish, it means plate. If you are good in your English, you will not have problem with biology at all. Petri dish means plate, Petri plate. In fact, sometimes we call, that's why you didn't call it plate count. You can see, so it's talking about the counting of the microorganism on top of the plate. So just like we have our own food inside plate, microorganisms also have their food in the plate. This is a bottle, this is not a plate. So the plate of the microorganism has a cover. We call the cover the lid. Then the plate is flat. It's not long like this, it's flat. So you put the food of the microorganism in the plate, you pour it out as the agar, nutrient agar or potato stress agar, depending on the type of microorganism. After you have put the food inside, the food will have been heated up. When you see molten, it's in liquid form, you pour it. When it's cold, it will solidify. So on top of that solidified agar, microorganism will now be put. Then you cover it. You don't cover like that, you just put it on it and then you incubate it. So after incubation, for bacteria after 48 and 24 hours, you will see the colonies growing. So these colonies are what you now count. That is why the first one method is plate count. So if you want to enumerate a bacterium or a fungal or any type of organism in a particular food sample, you can use plate count. You grow it, you can use settling plate. So you can't use settling plate because it's not air. You use settling plate method for air, microorganism in air. But in this case, you can use pour plate method, or you can use spread plate method for detecting the microorganism in the food. You can also use streak plate, where you streak and then you streak on the plate. So the colonies that develop, you count it. That is plate count. So the colonies will develop on the plate distinctly. They will grow separately. So each of the growths is is a convergence of different of thousands and millions of that organism in one place. So that is why you can see them on the plate. Normally, you cannot see microorganism with the unaided eye, but in this case, it is growing in millions at a spot. So that's why it will now become visible. So you count it on the plate. This one, all these are the plate, and that we call that one total plate count or plate count. Another method to enumerate or to see the number of microbes on a plate in a food sample is the most probable number. Most probable number counts. You do a series of tests with some test tubes. You do some dilutions, then you use your spec to check some things, to check the readings based on the dilution. Then you get 3 values for each food. Those 3 values are now compared with the MPN table. The MPN is the most probable number. MPN, most probable number. So you put the value you get, you put it and check it with the plate, then it will give you a particular value again. Then you will now know that, okay, that's why it's called it's not the accurate number, it's most probable. Based on the values you got from your own test, you now check that value on the most probable number table chart. Then you will now compare it, you will now give you a particular value. If you get this value for the first tube, this value, for the second tube, these are the 3 values you get. So in most probable number, just like the name MPN, there are 3 values. You also get 3 values for the MPN. So you now go to the MPN table, you check, then it will give you a particular value. Okay, if the first number is 2, second number is 1, the third number is 5. That means the most probable number in that organism is not 215. It will give you the value in the chart. It's not something that you memorize. I hope you get that. Then there's also the dye reduction test. Just like the name implies, the dye is reduced, and then you check by what is this dye reduced by this food sample, and that will tell you, let you know, these are standard tests that, okay, if so food sample is tested on so dye, and if you reduce the dye by so value, that means this is likely to be the amount of microbe in the food. Then we also have the electrical methods, where you use some electrical machines to check for the amount of bacteria in the food, the amount of microorganisms in the food. And one of the electrical methods is use of the spectrophotometer. You can use the spectrophotometer to check for the number, because spectrophotometry, we are still coming back to microbial growth. We'll talk about the methods of measuring microbial growth, which one of them is a spectrophotometer. So a spectrophotometer, we allow light to pass through a cuvette. Then, depending on how turbid the solution is, it will determine the amount of light that will pass through, and to determine the amount of reading that the spectrophotometer will read and give us the value. So we also have the ATP determination. ATP, if you remember, ATP is adenosine tri what? Adenosine tri what? ATP, adenosine tri, ATP, adenosine triphosphate. Thank you. Let me just ask this question. From your biology, what is the end product of glycolysis? What is the end product of glycolysis? What is the end product of glycolysis?
So they try to give people who don't know that means fine. So one point they say one point snack. I want you to buy the same snacks. Oh, ewa buy the same. Like small chicken. That my friend told me no to me lucky and lava. I swear, our meta. They can come in.
So let's go on. These people were just making a noise over there. So apart from ATP determination, you can also use immunological methods. Just like the name goes, immunology. In immunology involves antibody-antigen reaction. Just like when they do a blood group test, they collect your blood group, put it on a slide. If you have seen it before, on a slide, or on a tile, on a white tile, they put it in 4 places. Then they put the antigen A, antigen B, then antigen AB, then antigen O. Then they mix each of the antigen with your blood in the 4 places. So the one that reacts, that means there's the antigen that is reacting with the antibody. They'll be clumping. So they will know that that is present and they interpret it. So you can also use that to detect what you have in the food sample. Then you can also use the most recent one, which is the DNA RNA methodology. So we can also use the DNA RNA methodology. So that is just as that is. So let's further ahead now and look further at methods for the microbiological examination of foods. There is what is called the indicator organisms. Indicator organisms. What did I call them? What did I call it?
Indicator organisms.
Indicator organisms. Thank you. Now, in your department, how do you know when your HOD is around? You watch for his car. Not even HOD. You maybe, except your HOD is your level advisor. You will know if he's around when lecturers bring their car to school normally, before the strike, before the fuel wala. You will know that the lecturer is in school if you see his car. True or false? That's true. So the moment you see the car of the lecturer in his office, you know, So that car is an indicator that the lecturer is what is present. But there are some times that the car will not be there and the lecturer will be what? Will be in the office. So the indicator is just telling you that it's just an indicator. It doesn't mean the person is there. But in most cases, in 90-something%, once you see the indicator, you know that the man is in the office or the woman. Are you following? The same thing in a food sample. You can use an indicator species to know if an organism is present in a food, if a bacterium or a fungus or a virus is present in a food. The reason why we use indicator organisms is because most of the time it is difficult to test for the pathogen itself. Because pathogen is in the food. It's difficult to test for the pathogen or to test for the spoilage organism in the food. The detection process is difficult, it's laborious. But the detection process for the indicator is not laborious. I think you get it now. Most of the time, to test for the indicator is cheaper. But to test for the main organism causing harm in the food is expensive. So what you just do, just go and look for the indicator. Once you see the indicator, you tell them, don't eat this food again, don't eat this food again, don't eat the food until we complete the test. Let's say, for example, in a town where they have cholera, Vibrio cholerae. Now, you as a food engineer, you are, they have cholera in this town. What are you going to do? They just say, well, people are drinking this water, they are dying. Or they are eating this food, they are dying. Usually, they will not call the food engineer, but the food engineer should know about this also because he is the one that is the food engineer and the food is coming from what the machine has produced, or has manufactured for them. So they will call the medical doctor or call the microbiology, call the medical doctor, the medical doctor, call the microbiology, call the medical lab person, call the water microbiologist and say, what's happening? Call even anybody they can call the copper that I say is an engineer. Engineer, see what people are dying. What do? You have to tell them to what you just like that's last week we looked at it. They asked the individual or the man or the woman, the medical doctor asked for the history. Where did you go in the past 1 week? You can see. So where the person went to a party and ate food. Then the doctor suspected food poisoning. Food poisoning does not mean that somebody put something in the food. It can be that during the preparation, some things reacted together and then the food was affected. A lot of people may eat a food that is poison. Some may react to it, some may not react, depending on their immunity. We'll talk about all that too. Even those who are immune, who have good immunity, some may still react to food poisoning depending on their antibody antigen reaction and how they have been taking care of themselves from time. All this we spoke about last week. Now, coming back to the concept of indicator species, now instead of going ahead to look for Vibrio cholera, which may take a number of days, so instead of that, and then maybe in the place there's no microscope, so you look at it, you suspect this cholera that is happening. What can cause it is food and water. Ask them, which water are you drinking this town? The water is from that river. Okay. You tell them, okay, for the meantime, don't drink water from that river again. Which other water do you have? The water is far. Another place. So what you do as a food engineer is, there are a lot of things that will be done. Definitely the water is coming from up the river. So you can trace up, you can go with some guys in the town, up onto the river, you trace it. Where the water is coming from, you trek and be walking. By the time you get to the next town, next 2 towns, and you see where they are pouring excreta, pouring feces. When they pour feces from that town, it's coming down to this town. That is what is causing the cholera now. So there are 2 options. Either they should go far after where they pour the excreta to fetch their drinking water, or they should look for another source of water, and those people should stop pouring excreta there. That's what they call sanitary inspection or surveillance. In surveillance, you go through all the process by which the food or the water gets to the point where it is being taken. So, coming back to the concept of indicator organism, it is frequently necessary to conduct a microbiological examination of food to detect its quality. You must conduct a microbiological examination of food to detect its quality. But it's not in all cases. I told you that day, charcoal, Chicken Republic don't have microbiologists that is doing any the microbiological test or food test. But they have people that know about food hygiene, like we are talking now. That when you serve a customer, you use a particular tray to bring meat pie from the kitchen or from anywhere, and you put it on the floor. Before you use it to bring another one, you must wash it clean. So you know, so they use preventive measures in the food industry. It is in very big food industry that they do testing after production. But in those small food industry, they don't do any testing. What they also do is they ensure hygiene is maintained. And if hygiene can be maintained, I can tell you, there's no how the food will be contaminated. The food will not be contaminated. All through the line, until the food gets to the customer, they ensure proper hygiene. The food will not be contaminated. Where the food gets contaminated is where there is a break in the hygiene protocol along the food chain line. So it is frequently necessary to conduct a microbiological examination of food to determine its quality. This may be necessary to examine its shelf life, its suitability for human consumption, or to confirm that it meets some established microbiological criterion. The total mesophilic plate count is widely used as a broad indication of microbiological quality. That means the total mesophilic plate count, the total amount of bacteria that we found in normal temperature. Mesophilic organisms are organisms that from temperature of like 27 to like 40 something degrees Celsius. Then we have the thermophiles. Those ones, they grow at elevated temperatures. Then we have the saprophiles. Those ones grow at lower temperature. So most of the microbes we found around us are mesophiles. Their temperature is around just a little bit above ours or below or exactly. So those are mesophiles. That's why in the incubator, we incubate at 37 degrees Celsius for 24 hours for bacteria. So the total mesophilic plate count is widely used. I've mentioned that before when we talk about enumeration as a broad indication of microbiological quality, although it is unsuitable for this purpose in fermented foods, which contain large numbers of organisms as a natural consequence of their preparation. So you can see now, fermented food, you cannot use total mesophilic bacterial count. Because during the process of fermentation, what is fermenting the food? Okay, it's like this alcohol, ethanol you produce that day. You know, it's okay, you want to check the quality of the ethanol. You want to check the total fungal count. You want to check the total bacterial count. You are going to find a lot of yeast in the ethanol because yeast was used to ferment the ethanol. So since it's fermented, it will multiply. A lot of yeast will still be in the ethanol. So if you use that for fermented food, you are going to get a lot of microbes because microbes are involved in fermentation. And when they are involved, they won't go away, they'll still be in the food, but they are not harmful to humans. They're not harmful to health. They are normal flora of the body. And I told us last time that we have normal flora of the skin, normal flora of the mouth, normal flora of different parts, in different parts of all the parts of the body have their normal flora. Are you following? So this is going to be our class work for today, which I'm going to submit at the end. What is the normal flora of the intestine? What is the normal flora of the mouth? The bacteria, normal bacterial flora of the mouth, normal bacterial flora of the intestine, and then normal bacterial flora of the hands. I'm not coming anywhere again. So I've told us that in food that fermented food, you cannot use total mesophilic bacteria. You cannot use total bacterial count for fermented food. Why? Because the microbes are used for the fermentation, and definitely microbes will be many in the fermentation, in the fermented food, because microbes are involved, and as the process is going on, more microbes are involved. In other words, in other foods, knowledge of the total count may be useful, but it is often of more value to obtain an estimate of the numbers of a particular component of the total flora. Examples of this could be molds in a cereal, psychotrophic bacteria in a chilled food. You can see psychotrophic, write it down. Psychotrophic. We are still going to come to that later. Psychotrophic. Psychro, P-S-Y. C-H-R-O-T-R-O-P-H-I-C. Psychotrophic bacteria in chilled food. Chilled food has psychotrophic bacteria, meaning that they are in cold temperature. I told you that mesophilic bacteria are at our own body temperature, while psychophilic are in cold places. Then the thermophiles and the hyperthermophiles are at higher temperatures. Let me take the class work again. The normal bacteria flora, F-L-O-R-A, of the mouth. That's number one. Number two, normal bacterial flora of the stomach. That's number two. Number three, normal bacterial flora of the intestine. Intestine is different from stomach. When you write them, you see that they are different. Normal bacterial flora, number one, of the mouth. Number two, of the stomach. Number three, of the intestine. Number four, of the hands. Of the hands. Number five, of the skin. Of the skin. So that by the time you are shaking your friend, you know the normal flora that you are collecting. And then, okay, I saw of the mouth already, that's good. So that by the time you are talking, you know the normal flora that is coming out of your saliva. Because the mouth and saliva, they are interlinked together. That's why when some people are talking and they bring saliva and talking, people said 1 million bacteria rendered what? Homeless. Or if somebody sneeze, some people that know, they say that it has rendered 1 million bacteria homeless. You know, them homeless, they will be in the air or they land on the surface, they will go to new homes. They're not homeless, they are going to new homes. So if they are normally in the nose before, he sneezes them out, they find themselves on the floor. The friend that is there, some of them will land on the person's body too. And then people around too will take some by air. So they are not homeless. The 1 million bacteria will get new homes. So anaerobes in a vacuum-packed food or yeast in a fruit beverage. A quite different reason for a microbiological examination of a food may be to identify the cause of spoilage in the presence of a pathogen, where a food has been implicated in foodborne illnesses. You can see it say a food has been implicated. If the food is not implicated, you won't test the food. So you can see most times it is when the food is implicated, like charcoal, like Chicken Republic, like the place, like this joint inside the school. What was the name? Yeah, item 7. They won't do any food test, but they make sure that the food chain from buying to cooking to the water to everything is in order. You can see item 7, the way they serve you. They cover their nose, use mask in their hand. But the time that they will now start testing the food is when people suspect that people are falling sick or they're having diarrhea or something because they ate that food. So it is when a food is causing a problem that they will now start testing the food. That's why he's saying here that when a food is implicated, the 40 minutes for this Zoom may still go off, When it goes off, we will continue immediately after. I will not take any break. I will come up again immediately. So let's take note of that. So when a food is implicated, that is when you now start doing some tests. The isolation of specific pathogens, which may be present in very low but significant numbers in the presence of larger numbers of other organisms, often requires quite elaborate procedures. I've said that before. What is explained now is the concept of the indicator species. If you are going to go and check that your lab advisor is in the office, that means you will leave from come-sit area. You will now go to but from come-sit, you can still see the car of the lab advisor. It's there. But you didn't see the car. You have to now walk to the department outside and go inside. Then inside the building, you have to knock the door, open the door, and it's in there. Then you now go back again. But go back to where I find at come-sit. Whereas from come-sit, you can just say, the car is there. That means it's inside. The same thing with indicator species. Indicator with you will not allow you to make any trouble. But the organism itself will be problematic, that you can't just find the organism just like that. But the indicator will tell you that, once you see this indicator, that means the organism is there. Once the indicator is not there, that means the organism that caused the disease is not there. So that's what they are saying here. It may involve enrichment in media which encourage growth of the pathogen while representing the growth of the accompanying flora, followed by isolation on selective diagnostic media, and finally, the application of confirmatory tests. So all these are going to be done on the pathogen before you can detect that this is the pathogen. But indicator species, you don't need to do all that. Though microbiological criteria or the investigation of an outbreak of foodborne illnesses may often require the monitoring of certain products for specific pathogens, the difficulty associated with detecting low numbers of pathogens make it impracticable as a routine procedure to be applied without good cause. You can see it's expensive, it's laborious, the pathogen may be in small quantity and it's causing a disease. So you can't run after the pathogen, but the indicator are many. Once the pathogen is around, the indicator will be many. So you try to look for the indicator. An alternative to monitoring for specific pathogen is to look for an associated organism present in much larger numbers, and that is an indicator organism. This is a concept developed originally for pathogens spread by the fecal-oral route. Fecal-oral route. Fecal-oral route is from the feces to the mouth, and it's common in children. It's common in children that don't use pampers. That's why all these kind of child diseases is common in low-income earners. They cannot afford pampers. So when so doctors know all this. That's why you guys should work hard, study very well now, so that you can afford pampers for your children. If you can't afford pampers, you'll be buying, your wife will be using a cloth that you use before you wash it. The feces have done on the floor. The child have touched it and put it in the mouth again. So the everything is pampered. If you remove it like this, everything will pack together. Just use what do they call it? This thing, tissue paper that is will be cool like this again. You just wipe it. Use baby wipes. You wipe one, use another one. You wipe it, and then everything will pack together. You throw it away. Use another pamper straight. Everything is clean. But low-income earners cannot afford that. So that's what you are saying there. So an associated organism. So as I was saying, associated to this pathogen, that's why we found the concept of indicator species. A good indicator organism should always be present when the pathogen may be present. So a good indicator must be present when the pathogen is present. When the lab advisor is in the office, his car also must be present. If not for this fuel wahala in Nigeria now, the fuel price have gone up. So most lecturers bring their car twice or 3 times in a week. But when the fuel was not like that, every time you just see the lecturer's vehicle. So that's good indicator. Must be present. Every time the pathogen is present, indicator will be there. It should be present in relatively large numbers to facilitate its detection. To facilitate its detection, that means detection of the indicator organism. It should be present in relatively large numbers so that it can easily be seen without any problem. Then another thing is that this indicator organism should not proliferate in the environment, be monitored, and its survival should be similar to that of the pathogen. The indicator should not be just growing, that will not proliferate, should not be growing like that. Then the survival of the indicator species should be similar to that of the pathogen. Now, if a lecturer brings a car to school, if he's still in the school, will his car be in the school? No. If his lecturer is going home, will the car stay in the school and lecturer is at home? No. So the car is what brought the lecturer and what will take the lecturer back to the house. So if a lecturer brought his car and he parked his car, will he go and enter public transport or independent transport and go home and leave his car there? No. So anytime the lecturer is around, the car will be there. That is what the indicator species 2 is. Anytime the pathogen is available in the environment, the indicator will also be available. Escherichia coli is a natural component of the human gut. Can you hear that? Escherichia what? Escherichia coli. You can't miss it. It must come out. I must ask you a question about it. And if you miss the spelling, you miss it. If you miss the spelling, you miss it. If you miss the spelling of Escherichia coli, you miss it. Escherichia coli is a major indicator of fecal pollution. It's a major. It's not really a pathogen, but you can see from the description I'm giving you now. Escherichia coli is a natural component of the human gut flora, and its presence in the environment or in food generally implies some history of contamination of fecal origin. Any E. coli comes from the gut. That's from the gut, from the intestine, from the stomach. How will something that is in the stomach get to your food? That means from the gut, from the intestine, it gets to your feces. Then from your feces, it comes to your food. Anything that enters your food from feces is a contaminant. That's why I said oral fecal route the other time, that most of the things that cause disease are from the excreta. But E. coli comes from the excreta too, but it does not cause disease. It is an opportunistic pathogen. Normal E. coli. Now, if E. coli is found in food, it's an indicator that there's another contaminant from the feces that may be present inside of the food. Moment you find E. coli in your food, that means another contaminant from the feces may be present inside of your food. Inside of your food. And that is why you have to be very, very careful with the food. The moment you see E. coli, you don't, no story, just throw the food away. They should not take it again. The moment E. coli is there, it's an indicator of fecal pollution. It's not the pollutant, it's not the pathogen, but its presence shows that the pathogen is present. I think you get it now. Presence of the car, of the VC's car and VC's driver shows that the VC is around the place. I think you get it now. Presence of the driver of the VC and the VC's car in a place shows that the VC is around the place. The same thing, the presence of your HOD's car, labor advisor car, in the parking lot of the department shows that the HOD or labor advisor in his office. E. coli's presence in a food or in water shows that a pathogen is also present. So we don't bother looking for the pathogen the moment you have seen the indicator, which is E. coli. Hope you are following. Hope you are following. So, although E. coli usually grow Although E. coli usually grow in water, in temperate countries, it can grow in the richer environment provided by many foods. Although E. coli cannot usually grow in water in temperate countries, it can grow in the richer environment provided by many foods. In the tropics, like Nigeria, E. coli is found in water. It's even found in some soil. But that soil, you are sure, maybe somebody have defecated in the soil. The material I'm looking at here says that although E. coli, I'll take it again, so that, because I'm going to share this material with you later, so that you go through, say, although E. coli cannot usually grow in water in temperate countries, it can grow in the richer. This book is a book written by somebody in temperate countries. And that's why the work of the lecturer comes in, so that I can help the student to understand. So that not just saying E. coli cannot grow in water in temperate countries. But we are in Nigeria, in Africa, we are not in the temperate region, we are in the tropics. And E. coli is common. It's very, very common. In fact, the knowledge about E. coli that I know mostly is from water. Because I'm typically, majorly a water microbiologist. And we test for E. coli like no man business. You must just, in fact, if you don't test for E. coli in water, you are not, if you cannot test for E. coli using this thing, that means you don't know anything there. Because it's an indicator of fecal pollution. E. coli is an indicator of fecal pollution. E. coli is an indicator of fecal pollution. All these people joining the class and leaving the class and making noise, they're going to make me to change gear in the way I'm giving my lecture. All this is so I don't understand. If from next week, I will not be, anybody that comes late will not join the class again. So I will put this thing there. So I don't understand all this one. I'll be talking, somebody will be talking there and other things. So that is part of it on the concept of indicator species. And then it's not only E. coli that is an indicator. There are many indicators. In fact, there are some viruses that are indicator. There is a material called the concept of indicators, which is a whole book where we talk about the concept. Different people brought different arguments. Okay, can't we use streptococcus? Even streptococcus. Streptococcus is also an indicator. There are even some viruses, Clostridium perfringens is also an indicator because it's also found in the soil. And many other indicators of fecal pollution. So in that book, there was an argument. This one we argue about the organism and give us advantage and disadvantage. And then from everything, we discovered that E. coli is still the number one, is still the best. There's no two like E. coli. Now, from the table I have here, we have Citrobacter, we have Edwardiella, we have Enterobacter. Enterobacter, by the way, is the twin of E. coli. We have Erwinia, we have Escherichia, we have others like Klebsiella and many others that are predominantly fecal in origin. Then we have another group usually detected in coliform test. Citrobacter is detected, Enterobacter is detected, E. coli is detected, Klebsiella is detected. Those four are detected. Then typically enteropathogenic in humans, only two, that is Salmonella and Shigella. Out of these two, E. coli is not pathogenic, Salmonella is pathogenic, and then Shigella is pathogenic. Now, already from that, Shigella and the other one have been thrown out because they are pathogens. But E. coli is not a pathogen, so others are not pathogen, and they are found in the coliform test. They'll be taken up for the processing. Now, for your knowledge, know that there are many other organisms that can be used as indicator, but E. coli is still the best based on advantages and disadvantages. Now, let's look at direct examination. we are talking about microbiological laboratory methods of assessing microbiological status of different kinds of food. So you can use direct examination. They bring rice to you that this rice, so if you like say is what cause diarrhea or is what cause that person to be having stomach problem. So what do? Directly as they bring the rice like that, you examine the rice under the microscope in the laboratory. You say bring the rice, then you just put it in sterile distilled water, small sterile distilled water with sterile something, then you put it inside and you turn it together, you shake it inside test tube, then just take about 1 ml of the water or 0.5 ml, put it on the glass slide, and then put it under the microscope and look at it. What you see inside, if it's dancing jogo, and the microorganisms are moving up and down inside, and you see this microorganism is the shape of cholera now, these organisms that cause cholera. Then from there, vibro cholera. Then from there but that is not final examination. That's why it's called direct examination. You just look at it. In fact, the food itself, you can look at the food like this. You can smell the food. They bring the food, physical appearance of the food. How does it look? So it's not every time, even from the way the food appears to your eyes, you can say, this food, even though you have not tasted this food looks somehow. Those are direct examination. Then you can also smell the food. Then you can touch it. Normal rice, normal fried rice, you know how it will be now, but this fried rice, how is it like this? As you touch it, as you smell it, you know that something is wrong somewhere. So these are some ways to examine the food by direct examination. Now let's look at the cultural techniques. Cultural techniques of examining foods in the laboratory. There's also what is called cultural techniques. Although there is clearly a place for the direct examination of a food, and you agree with me, you can examine a food directly and you see that this food, look at how it is. Look at how it is. Fried rice. Fried rice now. Just like some of the students that are doing rubbish inside the class, going up and down, coming back. Students go time lecture the one day. First class, second class, third class, one baby have to submit assignment, one submit late again, one class, all those kind of things. You know that then all those ones will still be looking. A ní kan ṣe practical, àwọn méjì ló tún ṣe, àwọn mẹ́ta ló ṣe, àwọn mẹ́rin ló ṣe, àwọn tó kù ò ṣe, gbogbo ẹ̀ lárí nínú picture. Wọ́n be that direct examination now. Show you get it now. Direct examination. Lecture is going on. Students don't wọlé tún jáde. Wọ́n kàn wọ́n ilé wọn anything tó fẹ́ ṣe. That's the same thing in the food test they are doing. Cultural techniques is now the next thing you do after the direct examination. You now go on to cultural technique. You don't just say, direct examination, but cholera. Doesn't mean that the smell is what caused the cholera. No, you are just suspecting. You are suspecting that it may be possible that this food is contaminated. Yes, it's a suspicion. Is there microbe inside? Is there vibro cholera inside the food that is causing cholera? That is why you now have to do cultural technique and other technique. I hope you get me now. That's why you have to do other tests to detect and to know what the food contains and what it does not contain. Although there is a clear, excuse me, my laptop battery is let me plug it. Okay. So as I was saying, I've plugged it now. I said, although there is there is clearly a place for the direct examination of a food for microorganisms. A full microbiological examination usually requires that individual viable proper groups are encouraged to multiply in liquid media or on the surface or within the matrix of a medium solidifying with agar. So, yeah. You can look at direct on the plate, you see microorganism smell, all those things are direct. You still need to do the cultural. Cultural means you grow them on microbial culture. The culture can be in broth, that means in liquid culture. Some microbes are in liquid culture and some are in solid culture. Now, what is agar? A-G-A-R. Agar is a polysaccharide with several remarkable properties. Agar is just like starch. Now, if you have gari, gari is supposed to make eba. You now have hot water, but the gari is small, hot water is plenty. Will it become solid? It will not be, even no matter how the water is hot, up to 1000 degrees C, the gari will not become eba because the water is more than the gari. That is what happened in agar. When the starch is less in the medium, then you have a liquid medium because the starch is less. When you add less agar to the medium, nutrient is there, but there's no agar, so it will be liquid. But the moment inside that nutrient that is liquid, you add agar to it in the molten form, hot, you add agar. When it cools down, it will become agar. It will become nutrient agar, so it will become solidified, just like your gari and your water, hot water. If the gari is sufficient and the water is sufficient, by the time you put it together, you turn it, you allow it to cool down, you will not be able to turn it like that again. It has cooled, it has solidified. The same thing with the agar. So that is the work of agar. Agar is a solidifying agent, just to solidify the media where the microbes will grow, so that by the time it's set, you cannot see the microbes growing on top of the agar. So agar is a polysaccharide. Although it is a complex and valuable material and so on and so forth, I've explained all that. You don't need much of that for this class. Now, the formulation of a medium will depend not only on what group of organisms is being studied, but also on the overall purpose of the study. So you want to form a medium, just like you make a bar. Can you make a bar for a 1-year-old child, for a 6-month-old child? No, now. It's not a bar, that's their food. So different people have their food. Old people have their own food. Even you that are young people, some people now, Indomie they ate today. Some people ate rice, some people ate beans. Some of you, as you are, you cannot eat. You can't eat eba. Some of you don't like beans. So everybody has his own characteristics. So if you give people eba, after some time, something can happen. Whereas some people, is that eba that they like? The same thing with the microorganism. You prepare a medium that contains only a particular kind of food nutrient for the organism. Some organisms like that, they will grow in that area. Just like in the class now. Now in the class we have 21 or 17 students. Let me even take the attendance right away. So, as I was saying, in the class now, we have a particular number of students. Some people, if you ask them to come to class, it's like you want to cheat them. So, if we say that there's no lecture, it's like, they are not happy. So, some people are organizers are going home to town now. You see, organizers at Ukyodo. If we ask them to come to school, it's like, no, this type of organizer is what I want to be. This type is what I want to be pumping. The same thing with organisms. Some of you now, people that are going to become lecturers, already systematically you are preparing yourself. Even your classmates, they are knowing you already. They are calling you doctor, they are calling you a FICO, because you are going to become a researcher, you are going to become a scientist, an engineer that is going to be not just stopping at the level, you are going to go higher. So the same thing with the organisms. Some organisms like this, so they have been studied. So there are different food for different organisms. So we have different organisms have their own different kind of food. So if you prepare that kind of food and you put that kind of organism there, they will grow there. So there are some spoilage organisms in food or food pathogens in food. If you grow them on particular medium, they are the only ones that will grow. Others will not grow. And that is what we call selective medium. And that is what we use to grow E. coli. In the case of there's Salmonella, there's Shigella, they can cause infection. There's what is called Salmonella, Shigella, agar. So that agar, that food is only for Salmonella and Shigella. Yes, other organisms can grow. But if Salmonella and Shigella is present, there's a particular color that Salmonella will show. There's another color that Shigella will show. Can you see now? Others can show any kind of color they like. But Salmonella and Shigella, they've been tested. That Salmonella will show this color. Shigella, on this Salmonella Shigella agar, this is the color they will show. So that's cultural. They'll use that to grow them. For E. coli, if you don't know this one, I don't know what you know in this course. So E. coli, it is EMB agar you will use. EMB, eosin methylene blue agar. Eosin methylene blue agar. Eosin methylene blue agar is what you use to grow E. coli. When E. coli grows, if E. coli is present in that water, in that food, let's say it's rice, and then you put it. So because you in the food, you will not be looking for Vibrio cholera. Remember, it is the E. coli, it is the indicator you'll be looking for. Moment is because how will Vibrio cholera enter food if not from the feces? If Vibrio cholera enter food from feces and it's causing disease in people, that means E. coli too must be present. If the level of exercise is in the office, his car must be outside. I think you're getting the gist now. So we don't look for the level of exercise, we look for his car outside where he park it. The ones who see the car, we know he's in the office. Then we cool down, we palm down. So if you want to check a food for Vibrio or for contamination, you check for E. coli. For fecal contamination, you check for E. coli. On eosin methylene blue E. coli, when it grows and it appears on the plate, it will appear as greenish metallic sheen. Greenish metallic sheen on the plate. This, what I'm saying, basically affects the microbiology, but this is microbiology. Sorry, microbiology, but this is microbiology. And honestly, I don't know where any one of you will find himself tomorrow. So take this in seriously. No knowledge is lost anytime, anywhere. It is going to be a big plus for you. Don't think that it's only the engineering stuff you'll be doing. When you get to some places where I'm expecting you to get to, not the local champion level, the real places where they are playing this game, this engineering game, you will see that you will think you're a biologist by the time by things we know. So I'm trying not to just give you short this thing like the last set. The last will just say, okay, take it easy with them. But if you take it easy with them, when they get out, you will see that it's not that easy, man. So you have the capacity to know it. A teacher, when I was in secondary school, I was a science student. We were doing further maths, English, physics, chemistry, biology, science, economics, Yoruba language. The woman now, they brought her. She was teaching Yoruba literature. Yoruba. They now asked her to come and be teaching us in science, SS2. Literature Yoruba. Yoruba gone out, can't tipa ni. Why your literature Yoruba again? When she come to class, we will go out, we won't do it. Because we won't already start waiting WAEC now. But the woman has a job to keep. So she came to class that day. She said something that made us to start doing that literature Yoruba. She said that you need to science. Tí wọ́n sọ pé ẹ mọ̀wé. Ṣé wá sọ pé science, chemistry, physics, ó ti gba gbogbo ọpọlọ yín kò sáyè fún literature Yorùbá ń bẹ̀. That was what she said. All of us, the thing ginger ọ̀gá òótọ́ ni o, àwọn àtàmọ̀wé báyìí, literature Yorùbá náà agbúlọ̀ kò sáyè fún. That was how we started studying something. We started reading literature of Yoruba books. The literature in Yoruba, we come to class, we sit down, we read it, and we enjoy the classes. And it helped us later in life too. So even though you're engineers, there's space in your memory, your medulla oblongata and cerebellum for all this microbiology stuff. So if you're engineering students, it's a professional course, so microbiology should not be a big deal for you, I must say. So we have different kinds of media, general purpose media, selective media, and many other kind of media that are available. So you will see all this in the material we're going to give you, talking about the media and many other things. So moving on and trying to see what we want to do. So already I've told you about the enumeration methods. Number one, the enumeration method that I said is what? Enumeration method. That's what we started with. Number one was what?
Plate count.
Plate count. Thank you. You are following, man. So under enumeration method, we have the plate count. Plate count. The bacterium have grown on the plate. And as they have grown, you just come and count the colonies. Just count them. You've grown them on the media. Then what grow, you see it and you count it. That's what plate count is all about. So it has already been suggested that to count microorganisms in a food sample by direct microscopy has a limited sensitivity because of the very small sample size in the field of view and the magnification needed to see microorganisms, especially bacteria. In a normal routine laboratory, the most sensitive method of detecting the presence of a viable bacterium is to allow it to amplify itself to form a visible colony. This forms the basis of a traditional pour plate, spread plate, or mouse and mirror drop plate still widely used in microbiology laboratories. So that is what I've just explained that you're having here. Let me see if we can go to the next one. The next one is the most probable number count, MPN. An alternative method of enumerating low numbers of viable microorganisms is that referred to as the most probable number method. The method is usually based on inoculating replicate tubes of an appropriate liquid medium, usually 3, 4, or 5, with 3 different sample sizes or dilutions of the material to be studied, e.g., 10 gram, 1 gram, 0.1 gram. The medium used has to be designed to make it possible to decide whether growth or no growth has occurred, and the number of positives at each sample size or dilution is determined after incubating the tubes. The MPN is obtained by referring to a table such as that shown in table 10.4. I will still give you the material. There are computer programs for generating MPN values from different designs of the experiment. I will explain MPN already before I get into this place. From different designs, and these programs can also provide confidence limits for the MPN and suggest what the likelihood of a particular combination of positive results should be. A modern variation on the MPN scheme is the use of hydrophobic grid membrane filter. Hydrophobic grid membrane filter. Honestly, if you are going to be using some of these things in the real laboratory, in the industry, it is this hydrophobic grid membrane filter they are going to use. They're not going to use MPN again. But this HGMF hydrophobic grid membrane filter is based on the MPN principle. So that means you have to know the MPN. If you don't understand the MPN, you just not get the HGMF. A sample is filtered through the hydrophobic grid membrane filter, which is divided by a hydrophobic grid into a number, normally 1006, of small cells or growth compartments. After incubation of the filter on an appropriate medium, each of these cells is scored for growth or no growth. This can be done either manually or automatically, and the count in the original sample determined as equivalent to a single dilution MPN using 1600 tubes. So, one application of the MPN which allows one to calculate the maximum number of organisms in a batch of material is based on 2 class attributes sampling plan. If a number of equal size samples is taken from a batch of material and all shown to be negative for a particular organism, then the maximum percentage D, there's a formula here which you're going to see, and all these are mathematics. I think you will enjoy it when you get to that point. So let's look at the alternative method. We have the direct method, we have the visual, we have the microscopy, then we have the cultural method, then we have the MPN. You can see there are different methods. So I'm telling them now, if I don't get to exam or test, they say, okay, well, list and explain the method used for this. Don't say you don't know it. We'll be rounding up in 10 minutes. Sure, you understand. If I don't say get to exam now, say list and explain the methods used. You can't say you didn't get it. So it shouldn't be that way. Okay? So alternative methods now. I'm not sure we complete this today. All right, so let's go on. Cultural methods are relatively labor intensive and require time for adequate growth to occur. Many food microbiologists also consider that the traditional elimination methods are not only too slow but lead to an overdependence on the significance of numbers of colony-forming units. Food manufacturers require information about the microbiological quality of commodities and raw materials rapidly, and it could be argued that an assessment of microbial quality is as important as a knowledge of numbers. A number of methods have been developed which aim to give answers more quickly and hence are often referred to as rapid method. What did I call it? Rapid method. What did I call it? Rapid methods. Can you say that?
Rapid method.
Rapid method. So we have only one person in the class. It's okay? Problem. So now, these alternative methods can also be called rapid methods. Can you show you? If you don't come to class, you won't. The kind of question I'm going to ask is not going to be direct. I'm telling you, everything I'm going to ask in the test or exam, I'm already saying it. Because the alternative method also called rapid method. Now, if I just reading, you just look, and that's okay. Explain the rapid methods. Do you not say rapid method are the method that make it to be fast? Is that rapid method? Rapid method are the alternative method. Yeah, make it to be fast. But that's the alternative method. Then you not talk about the alternative methods. But someone that didn't come to class, I say, tell me about the rapid method. You just say rapid method are the method of food sample that is rapid. Method of food sample that is fast. Is that the rapid method? No. Rapid methods, yes, you need to be fast. You have described it. And they also call alternative methods. And some of the methods are what? Dye reduction test. Because you can see the explanation I've just given you for the alternative methods. The main methods are direct, MPN, and cultural. Cultural, I told you that bacteria takes how many hours to grow? How many hours the bacteria takes to grow? I'm waiting for you. How many hours does it take a bacterium to grow? It takes 24 hours. And you are in a lab, in a food processing company. You are in Cadbury, you are in Guinness, you are in Nigerian breweries, you are in international breweries, you are in Nestle Foods, you are in Cadbury Nigeria Limited. Another food company where they make Maggi, where they make spaghetti, where they make different kind of foods. And you are there and you want to check after the production, after that line, after that batch of production, you want to check whether there is E. coli there. Then you have to wait for 24 hours. Does that mean after every batch you have to wait for 24 hours before the next batch? So what will the workers be doing before the tests come out? That is why the rapid tests come out. That is why the alternative tests, also called rapid tests, now come. Which number one of them is a dye reduction test. Very rapid. Just take something from the food you have gotten, add it to it. Immediately the result will come out. And it's still based on the cultural methods, on the knowledge. The next one, apart from that, is electrical methods. So when you plug some things, you get it, some chemical things will come out, you read your score there immediately, and you go on with that. My data is used up, but let's see. Then the next one you have is the, apart from the electrical method, I've mentioned it. I will send the material to you so you go over it and you look at it. I will send it by email to one of you because I'm not sure the class is around. Then the ATP determination, I've said it before, so it's also here. You read over it. All these are rapid methods. Then you also have the apart from ATP, you have the others. Then we have the immunological methods, which are also called rapid again. So we look at that, and then DNA, RNA is also there. So these are what we're going to stop for today, and we'll continue in the next week. We'll continue. So send in the task I gave you in the next few minutes. Then before I go, I would like to add one to it. And what I want to add is the name of the organism that is a major indicator of fecal pollution. In addition to the other that I've asked to do before, the name of the organism that is a major indicator of fecal pollution. So you add that one to it and send it to me. So I've told you the microflora of the mouth, normal flora, normal bacteria, not fungi, not algae, bacteria flora of the mouth, of the stomach, of the intestine, of the hands, and of the skin. That's 5. So you put them separately. It cannot be one organism. It can be 1, 2, 3, 4, 5 bacteria. Just name them. Just put them there. Then for fungi, not fungi now, for the stomach, for the intestine, for the hands, and for the skin. Then now the name of the organism that is used, or that is a fecal indicator of fecal pollution. You tell me the name of the organism. So that will be everything will be 6. So we'll see you next week. I'll be expecting the answers in the next 10 minutes. Please, if you tell those who are not in class not to send it. If they send it, I will take it. I won't take it easy with anyone that does that. And if you don't submit in 10 minutes time, the time now is exactly this. If you don't submit in 10 minutes time, that will be all.